Hello and welcome to this video podcast for human genetics. This is going to be the first of a few podcasts where we're going to begin our discussion of population genetics and evolution. Before we begin though, let's think a little bit about where we've been. Essentially, we've been studying the individual. In general, we've thought about how this individual might have a genotype. They might have a genotype of little a, or big A, or big A. And we'll say that little a provides the trait of albinism. So we understood how these alleles and in these combinations, how these alleles could affect this individual. We also took up the discussion of how he or she might pass this on to the next generation. So let's say this individual had the genotype of big A, and they had children with somebody who had the genotype of big A. We understood that they would have 25% chance of having a child with albinism, these individuals here. So at this level, the individual level, and thinking about passing on to the next generation, we've discussed quite a bit. But we know that we as individuals, we don't live in isolation. Most organisms, humans included, live within populations. So what happens if we branch this out and we consider this person here who's now in this population with many other people? And these individuals in this population, they're each going to have two A alleles, say in this case. So they might have some individuals who are big A. There might be some who are big A, little a. But there also might be third alleles. Maybe there's somebody who has a different allele, and we'll call it A1, and maybe another one called A2. Maybe A1 with little a, A2 with big A. A population is going to have all possible alleles in it, not just the two that are possible in any given individual. So let's begin here by defining a couple terms. In particular, let's think about the term population. Population is best described as a group of individuals of the same species live in the same area, meaning that they could mate with each other. So what I'd like to do for the rest of the podcast, we're going to kind of leave much of the individual aspect behind and focus on populations and how we're going to study populations and why it's important to study these populations. So let's define a few other terms then to help us as we move forward. We probably should define the general term here of population genetics. As described here, we have a population and we have all these different alleles in this population. So population genetics is the study of all alleles in a population. We should also define what a gene pool is, because that's related. We can think of the gene pool as all the alleles in a population. And down here, let's define gene flow. This is also important, and we'll talk about this as we go forward. But gene flow is the movement of alleles between populations, usually caused by migration and mating. We would expect that for most populations, the gene pool will change some due to many factors. One of those factors is gene flow. As we move deeper into population genetics, it's also important to define what we mean by phenotype frequencies, genotype frequencies, and allele frequencies. Essentially, phenotype frequency is based on observations. That is, how common is the trait within a given population. So for instance, your book talks about the disease PKU. And we know people from, say, Turkey, it's pretty common. 1 in 2,600 individuals. People from Japan, however, have a much lower frequency, about 1 in 119,000. So population geneticists will study phenotype frequencies to help make predictions about how likely it is that an individual will have a particular trait. Genotype frequencies are the proportion of heterozygotes and homozygotes in a population. So if the gene P is linked to PKU, you might be interested in knowing the percentage of individuals who are homozygous dominant, those who are heterozygous, big P, little p, and those who are homozygous recessive, little p. This gives you a slightly more accurate picture than the phenotype frequency. This up here, phenotype frequency, is going to lump big P, and big P, little p together. But you really need to break this down because you want to know these individuals here because they're going to be able to pass it on. They're unaffected, but they can pass on the trait. Whereas this individual here, big P, can't. Little p, of course, can as well. So it's important to maybe start with the phenotype frequency and then you start to fine-tune and determine what the genotype frequency is. And as you might guess, the allele frequency is the proportion of each individual allele. And so let's work a small problem here concerning going from genotype frequency to allele frequency. So let's work a problem looking at those three terms there. Let's say we have 500 individuals in a population. 100 of those individuals have PKU, or you could say they have little p. So the phenotypic ratio is 100 over 500 PKU, individuals with PKU, 1/5 of the population. On an exam, you might report it as 100 over 500. If you have that option, 1/5 would be fine, or 20%. So that's our phenotypic ratio. We just talked about phenotypic ratios, and let's consider genotypic ratios. We can get some information from phenotypic ratios about our genotypic ratios, but not everything. For instance, we know from this problem here that we have 100 individuals out of the 500. 100 of those individuals are going to be little p, because we know that there are 100 individuals with PKU. Now, the individuals who are big P, now the individuals who are big P, or big P, little p, we don't know. We know it's going to add up to 500, but we don't know which is which based upon this problem here. So in a problem like this, I would either have to tell you what they are, or I'd give you information so you could derive it. I'm nearly certain I would just tell you. So let me do that here. Of the 400 individuals left, let's just say 250 of them are big P, and then 150 of them are heterozygous big P, little p. So the genotypic ratio for big P individuals is going to be 250 over 500. Heterozygous genotypes would be 150 over 500. And then the ratio of little p genotypes is 100 over 500. Then that leads us to our next calculation, which is allele frequencies. And as I wrote allele frequencies, I realized that I was changing some terms around, and so I should be consistent and call these genotypic frequencies. And up here, we should call these phenotypic frequencies. So allele frequencies. So with allele frequencies, essentially what we want to know is the frequency of big P alleles and the frequency of little p alleles in this population. So let's calculate those. Homozygous dominant individuals, each individual contributes 2 big P alleles. So if there are 250 big P individuals, that means we have 250 plus 250 big P alleles, because that's what these individuals, the homozygous dominant individuals, contribute to the population. The big P, little p individuals, they each contribute 1 big P allele. They also contribute a little p allele, which we'll get to in a moment, but we don't multiply this by 2 because they only contribute one big P allele. The little p individuals don't contribute any big P allele, so we can put a big 0 there. We have 650 big P alleles. Now what are we going to divide that by? Well, we don't divide it by 500 because we have more than 500 alleles. We have 500 individuals. Each individual provides 2 alleles. So as a result, we have 1,000 alleles. So the frequency of big P is 650 divided by 1,000. The frequency of little p is going to be calculated the same. Homozygous dominant individuals don't contribute a little p allele to the population. Heterozygous individuals contribute 1 little p allele per individual, or per heterozygote. And then down here we have 100 little p genotypes in this population. Each of these individuals will contribute 2 little p alleles. So we have to add 200 little p alleles. And if we add that up, we end up with 350 divided by 1,000. If you are asked on a quiz, a group activity, or an exam, you will be asked to do one or all of these calculations. And the math won't be much more complicated than this. We'll talk quite a bit over the next several whiteboards here about how allele frequencies in a population rarely are constant. They change quite frequently, in fact. And I want to say something about evolution. What we know is that the changing of allele frequencies, and we'll talk later about the factors that can cause these changes, but changing this allele frequency will lead to changes in genotype frequencies, and that will ultimately lead to changes in phenotypic frequencies. But it all starts with the changing of allele frequencies. If you start having a few more little a's in this population or a few more big A's in this population, that can lead to changes in the number of homozygous dominant, homozygous recessives, or heterozygous individuals. And we call this change in allele frequency, this is known as microevolution. Again, changing of allele frequencies in a population is referred to as microevolution. These are very small incremental steps. Ultimately, what's going to happen is through these small changes, so several rounds of microevolution, after they accumulate, what you can get is something we call speciation. New species form. And when new species form, that whole process we're going to call macroevolution. When we see big evolutionary changes, it really starts at the population level. And it starts with changing allele frequencies. And over time, those small incremental changes build up and can lead to speciation or macroevolution. Now what I want to do is think about factors that can change allele frequencies within a population that are these steps of microevolution. And I'm going to list 5 of them here now. So these are factors that cause allele frequency changes. The first we're going to call non-random matings. If it's a large population and the matings are random, one would not expect the allele frequency to change. But when matings are non-random, it can lead to allele changes. Okay, number 2 is migration. As individuals move from one population to another, that can change allele frequencies. The next one is something we call genetic drift. Genetic drift I've always found to be a difficult concept to comprehend. And basically what we mean by genetic drift is a random fluctuation in allele frequencies from one generation to the next caused by chance. And we'll spend a little bit more time with this later on, but for now just know that allele frequencies can change simply because of chance. Fairly unpredictable and it's fairly random. Number 4 here is mutations. Mutations are important as they can change the allele frequency by sometimes introducing new alleles or making alleles more favorable or less favorable. And number 5 is natural selection. Maybe little a genotype, even though it's homozygous recessive, maybe it is more favorable. And if it's more favorable in a population, that means it's going to have a greater chance of mating. And if it has a greater chance of mating, it has a greater chance of spreading little a alleles within that population. So natural selection is an incredibly important part of how allele frequencies will change. Well, that brings us to the close of this first video podcast on population genetics and evolution. In this podcast, we introduced the basic terminology and the ideas of population genetics. In the next podcast, we're going to talk about the Hardy-Weinberg equilibrium and how that explains an ideal state. If you have any questions at all over this material, please make sure you let me know. If not, I'll see you on the next podcast. Bye for now.